Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, part of the Finding Genius Foundation. I have my guest today is Christos Chinopoulos. He's at Semmelweis University in Hungary. He's an associate professor. He's a project manager of the RPPA facility, which we'll get into, and we're going to talk about cancer and cancer metabolism. So, Christos, thank you for coming.
Thank you. Nice to be here with you.
Yeah, tell me, what is the RPPA, first of all, and then tell me about your research, please.
Okay, RPPA stands for Reverse Phase Protein Array. And for those who are familiar with the technique of Western blotting, it is like Western blotting on steroids. And to put it in very simple words, it is able to kind of semi-quantify expression of several hundreds of proteins for thousands of samples. And this can be applied on solid tumors or healthy material. And several proteins on a single pathway can be examined at the same time. And it can give you semi-quantitative expression where the proteins are up or down regulated. And as an extension of this, you can have an idea whether this pathway is important in cancer metabolism or not.
Oh, interesting. So you can tell the level of regulation of these proteins. Is that what you're saying with this array?
You can say the level of expression and whether this is up or down regulation. Yes, that can be deduced later on.
If you do this multiple times, will it tell you, does it lead back to epigenetic marks? And are they the cause of the up or down regulation of the protein expression?
You can go after the reasoning why you have up or down regulation. That is one of the things that can be done after RPPA. Or what you can do is that if you see a pathway, for example, is upregulated in cancer, then you can try to find an inhibitor for this pathway, or you can claim that this pathway is important for cancer but not for healthy tissue. You have several targets amenable to chemotherapeutic intervention pretty soon, right after an RPPA run.
So this works on solid tumors, or does it work on blood as well?
It can work on blood as well, but our facility is made to accept exclusively solid tumor material.
So what, a biopsy will be done, or after a resection or a surgery, and then
Yes.
You'll swab the tumor, or you will immerse it in a fluid? Yeah. I mean, how do you get the information into the array?
Okay, so the workflow is that there is always a surgery, and then by law, the tumor material ends up in the pathology department, for which they are going to categorize and classify the tumor. And then in many circumstances, small parts of the tumors are given to different facilities for diagnostic or research. So in the vast majority of cases, we get a part of the tumor, and in most of the cases, we also get surrounding healthy tissue as well. We usually get 20 to 200 milligrams, so it's a very, very small amount of tumor that we get.
Okay, so how do you test the tumor material and also the healthy tissue? Is it composed typically of the same organ or same tissue type, and do you test that as well for comparison?
Yes. So it is very important to compare it to something, and the best is if we have healthy tissue from the same patient for comparisons. It's not always the case. Especially, for example, if it is a very critical organ, let's say for brain, it's very rare that we have corresponding healthy material. But if it is a breast or a kidney or liver, in many circumstances, we also have a healthy material together with the tumor tissue. But we always get those from the pathology institute. So the tumor has already been classified. I mean,
Okay.
The classification of tumor is already known.
So what kind of information do you learn from analyzing these tumors for their protein expression?
Well, we have, of course, hypotheses that we are testing. We are particularly interested in the metabolic features of tumors, and we look for proteins participating in biochemical pathways which are important for tumor survival during adverse microenvironments. For example, many tumors, they grow up during anoxic conditions or in the presence of several mitochondrial DNA mutations. and they alter their biochemical pathways to accommodate for that. So we are interested in those proteins that form these pathways. And these pathways can be literally hundreds of them. Each tumor may employ a different biochemical pathway for harnessing energy. And we are looking into that, which tumor is depending on which type of pathway.
Do you look at primary tumors only or metastases? And do you compare metastases versus primary tumor? Like, do you typically get in a sample both or just one?
Typically we get one, but sometimes we get recurrent from the same patient. And in these cases is also metastasis. The capacity of the RPPA facility is to serve the needs of approximately 50 million people. So this is usually for 6 or 7 countries. Yeah, it's not for a hospital or 2 hospitals.
Well, the reason why I ask is that the microenvironment of the primary tumor, it'll be, so the tumor itself will be composed of that tissue, the primary typically, but the metastases will be a very, it could be probably a more hostile microenvironment because, you may have breast cancer cells that are metastasizing in the liver, and now it's the environment by definition is very different. So that's why I asked if you look at primary versus metastases for the protein expression.
That is true. There are huge differences between primary and metastatic tumors, and even within tumors themselves, you find cell-to-cell specific differences. So each tumor cell is almost different than another. It's not homogeneous as a tissue. That is actually a major drawback in cancer research, that the so-called tumor heterogeneity. Almost every cell is different.
When you get the tumor material, is it all from one part of the tumor? Like if I imagine a 2-centimeter diameter tumor and I just picture it as a sphere, do you get one part of the tumor or do you get kind of a mixture of material from all over the tumor that was just scraped together and given to you by pathology?
We get one part of the tumor. Unfortunately, the tumor collection is a weak part of the RPPA because it usually depends on the surgeon or somebody in the pathology department who doesn't really care of what happens with the tissue downwards. So we get material many times which is not suitable for RPPA because it's too far on the edge of the tumor or it is too much in the center of the tumor, which is necrotizing. But, power in the numbers, when we get hundreds or thousands of samples, in the end, we end up with a sizable amount that we can work with.
Well, what do you notice? Like when you get a sample, do you know where it came from in the tumor? Do they have imagery and show you, I guess you call it the provenance, where it comes from? Or is it just they give it to you and you have to see if it works or not?
We only know is where it came from. And then we get a number which corresponds to the patient, but this is all encrypted in the end. So I only get to see, how does the tumor look like, and after I homogenize it to what is the protein and lipid composition. That's all. But the tumor
I don't know if they'll do it for you, but you don't have to know the patient's name or anything. But if they did an image of before they took out the tumor and then put an arrow to where on the tumor they give you the material and send that to you, that might help a lot.
Yes, that can happen because I can decrypt legally, of course, where did it come from. And then by knowing the social security of the patient, then I can track the progress, what kind of examination did the patient have, and I can see if they did anything regarding imaging of the tumor or any kind of adjuvant, let's say, blood work that can give you more information.
So what have you noticed? What are some of the correlations that you've observed so far? How many have you looked at? How many tumors?
So far, we focus on breast, kidney, and brain. And what I can tell you is that we have from a big project that's going on, we are trying to correlate the effect of mutations on known mutations on breast tumors that they have on the protein expression level of the same proteins. And what we see is that it's an extreme heterogeneity, really extreme heterogeneity. You cannot really deduce that there is one particular type of tumor with with a phenotype that it is that 60 or 70% of the population have it from those, of course, that they harbor a tumor. This heterogeneity is very, very striking.
Well, is there heterogeneity also in the protein expression, or is it just the mutations? I would guess there would be just the mutations in the cells cause it, right?
Yes, it's on both.
All right. So, what kind of patterns have you observed? Like, I don't know. Have you? Well, another question: have you looked at tumors pre and post chemo? Because I would think that would up the heterogeneity of the tumor after chemo.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
We don't have sufficient material of the same patient before and after chemotherapy, but what is known in the literature is that after chemotherapy, the protein expression and gene expression changes very much.
Well, again, what are some of the things that you have observed? What are patterns? What insights have you gotten? Like how many samples have you done? I'm sure quite a bit, and you've got to have some insights.
Approximately 3,000 samples we have done. We actually have got more insight regarding bioenergetic preference, what type of, food does the tumor prefer over healthy tissue, and the heterogeneity, what I just mentioned. It's very preliminary, but it seems like that many tumors, and this is concurrent with the existing literature, they prefer those type of substrates which are available from the immediate environment, from the surrounding healthy tissue. I don't know if you are familiar with amino acid glutamine, for example.
Yeah, I've heard of glutamine, yeah. So what's the function that you've observed, and what happens when it gets up or down regulated?
So we see both on the protein and also in the genetic expression level that there is a huge upregulation of the proteins responsible for the catabolism of glutamine in many tumors.
Yeah, because I've heard tumors, they don't use oxidative phosphorylation. They use like a fermentation process.
That's true.
So I guess they're very heavily glucose and glutamine dependent, right?
That's true. The glucose consumption is very, very high in tumors, but it's not for the purpose of energetics. It's because during the process of glycolysis, many of the substrates, they diverge into anabolic metabolism. But the pathway of glutamine catabolism is very important from the bioenergetic point of view for the tumor, because it does not need to follow oxidative phosphorylation. It can go through fermentation pathways.
But you said the glucose is not used for energy. It's used for what?
For anabolic metabolism, because during glycolysis, several metabolites diverge. They go into side pathways for forming material which are important for the formation of new cells, like glycolipids and sphingolipids.
Oh, so it's more diverted towards building new material. That's the anabolic component. But do they exist in a lower energetic state? Or is it glutamine takes the place of glucose somehow to help them produce energy? Like what is the main driver of a cell's energy that's a cancer cell?
It's for sure glutamine and some other amino acids, not glucose. They are not of the same energy state. They do not require the same extent of energy like highly, let's say like neurons that they fire up all the time, or cardiomyocytes. They harness sufficient amount of energy from fermentation processes for growth and metastasis.
So if someone had a very low glucose diet or a low sugar diet, I guess what we would expect to see is that tumor growth would slow tremendously and maybe even stop. But the tumors themselves, the cancer cells themselves wouldn't necessarily die because they're using glutamine essentially as fuel to keep them alive.
Yeah, there is a correlation between low glucose consumption and diminished growth rate. And this is actually the basis of the ketogenic diet. Unfortunately, tumor material is not possible to decrease blood glucose concentration to the extent that you can stop or reverse tumor growth, because the tumor requires very little. I don't think you can go into such a starvation state that you can decrease blood glucose concentration to such an extent, but it definitely helps. And it will also help very much if you diminish the glutamine provision to the tumor as well. These two will have a very good effect, diminishing tumor growth and metastasis.
How do normal cells use glutamine and glucose versus cancer cells? Like, what's the difference?
If you like this podcast, Please click the link in the description to subscribe and review us on iTunes.
Well, normal cells, there are some cells that rely exclusively on glucose, for example, like red blood cells, or there are other cells which rely very heavily on glutamine, for example, T cells. So there isn't really a rule of thumb that you can say about the healthy cells. Some need glucose, some need glutamine, some need both, but not to the extent that the cancer cells need. So the cancer cells utilize glucose and glutamine like there is no tomorrow. So if you were able to inhibit the provision of glucose and glutamine simultaneously, this would be detrimental to tumor survival.
So are you saying cancer cells are a lot more frugal with their use of resources, or it's just different than normal cells?
Well, what I can tell you is that cancer cells depend on glucose and glutamine consumption. That is for sure. Some other amino acids as well. They cannot survive without glucose and glutamine. They will be able to enter a sleeping state without glucose and glutamine. But perhaps not all of the cancer cells will die, but it would be impossible for having growth and metastasis without glucose and glutamine at the same time.
So going back to your sampling, you said that in some cases you've been able to get a tumor sample from someone and then they had a recurrence and then you sampled again. What was observed there in the same person with the recurrence? What was the difference?
Yeah, and unfortunately, these are not that many samples, and we are mostly focusing on mutations that lead to the recurrence of the tumor, which in most of the cases means that the resistance to chemotherapy has been developed. So I can't tell you more about the protein expression for these particular samples because there are very many and they are not diverted into looking into protein expression on the pathways that we are investigating.
Do you have a choice in the samples you get? Can you say, hey, we need more, tumors that have been hit with chemo previously? You just take what comes.
We take what comes because our capacity, now we work at about 5% of the total capacity. I just take what comes. I don't have priority list. I do not have a choice.
Well, if you're serving so many places, it sounds like you would be flooded at any one time with samples, or is it hard to get samples, or you have too many?
It's actually easy to get samples, but people are not really willing to give samples because this is a very new technique. The RPPA is a very new technique, and people are stuck into the genetic basis of tumors, so they would rather rely on research on genes rather than on proteins and metabolism. So we don't get as many samples as we would like, and we are definitely not flooding with samples, even though it's in the order of thousands. We can easily scale it up 10 times over what we are doing right, easily.
What samples do you want? What are you looking for that you're not getting very much of? And, how can you push to get more of those samples? Can you take them from, the U.S. or other countries? Can you expand your,
yes, of course.
Your advertising and say, hey, anywhere we want them, this is what we want.
Yes. We have a lot of samples from Russia. We have some samples from Austria. Before this pandemic, I was about to receive some samples from Greece and Turkey as well, but things got a bit hectic now. The point, I would like to get as many samples as possible from Central and Eastern Europe because these are closer. There is no RPPA facility in Central or Eastern Europe. There are only 7 RPPA facilities in whole Europe. So I would like to take care of, let's say, Poland, Romania, Slovakia, Slovenia, Hungary. But for now, I just have mostly from Russia, Austria, and hopefully when this thing will be over, from Greece and Turkey as well. It's very easy to expand because the sample that we need, it doesn't need to be very fresh. It has to be between 2 and 6 hours after excision. It can be frozen at -20, so no dry ice is required, and can be sent overnight. It's not such a big deal to send and receive such.
Are you allowed to solicit countries for samples? And have you, or are you just sticking to certain countries in Europe?
You need to have, of course, ethical permissions in place, and we do have them. And those people who provide the sample, they have to have permission as well. These are doable things. It will take, of course, some paperwork, some administration, and yes, it's possible to do this. Yes.
I wonder if you could do a bounty. Hey, these kind of samples we really want. We'll pay you more if you do it. I don't know.
Yeah, there is no money involved in this. The whole project is not monetized.
Well, so what would be some ideal samples that you want? What would you love to get that you're not getting enough of right now? What hypotheses do you want to test?
Okay, so I'm actually in very big need of more healthy material, and what I would have very much liked to do is collaborate with a team in Hungary. So in Hungary, you have a lot of body donors, okay? And whenever someone is dying, someone is dead and he's a body donor, then the team of doctors, they go there and they harvest all the tissues to be harvested. And I would like very much to join them, because then I can get access to fresh, healthy tissue material, because these people are, let's say, clinically dead, but not dead yet. So it would be possible to obtain materials for them, and they would serve as healthy controls, because a healthy control is as easily important as a tumor sample. Unfortunately, it's much easier to get tumor samples than healthy controls.
If a sample is frozen, how long will it last? Can it go for years? If it's deep frozen, let's say minus 80?
Oh, yeah, 40 years. It should not be placed into any fixatives. If it's placed into any fixatives, then it's unusable. But minus 80, yeah, it can go for years.
Okay, well, again, so what hypotheses do you want to test?
I wish to test a hypothesis that the metabolic pathways that we investigate, which is a small part of the glutamine catabolism pathway, is critical for tumor survival. And we aim to quantify the expression of proteins participating in that particular sub-pathway of glutamine catabolism.
Are there, I guess, at least in the U.S., we have health insurance and stuff. When you get samples, are there certain ones that pathology has very little interest in beyond the baseline, and therefore you can get more material, or you can get material from multiple sites?
I haven't got into this. I haven't faced that here in Hungary, or from the samples that we receive from Russia or other countries.
If you get samples from one person and you got primary tumor, 4 or 5 different sites in the same tumor, that would tell you something. And if you also got like a piece of a metastases in the same person, even if it's later on, that would tell you even more. I mean, with fewer individual people, but more samples per person, you might be able to see a lot more.
Yes, that's true. And that is the case with some ovarian tumors, because they tend to metastasize very much. But we have like 30 or 40 from those. So this is very little to have any statistical analysis. Unless I have 400 or 500 samples, then we don't really do any analysis. It's too few.
Well, really, how many do you need in order to, a cohort in a lot of experiments is very few. It could be 50, 70, 100. I mean, if you have several hundred of something, that seems like a pretty good sample size.
Yeah, so the rule of thumb is below 150. It doesn't make any sense to do RPPA. And the optimum is around 500, and not to go above 800.
Well, what's wrong with going above 800, by the way?
It reaches some limitations of the assay, and so it's not. So when I don't want to go much into the technique of the RPPA, but if you, at least for the machines that we have, if you go above 800, you have to have too many controls. Let's say you have to do things in batches, and you have to overlap to have many controls. That starts to be a bit too much work.
Well, so I don't know. Are you close to getting enough of a sample size to test certain hypotheses? Like what do you expect to figure out in the next year or so?
Yes, we have enough samples to test the hypothesis from the breast and kidney, and pretty soon from the brain as well. And we have actually already started in testing the hypothesis, which is corroborating studies that we do with cancer cell lines in cultures. I hope within the next year, within 2021 or maybe the next year, I will have some concrete data.
Okay. Are you observing in any of your samples that there's bacteria or other type of organisms that are kind of confounding the sample? Are you picking up any protein expression from microbiome related stuff?
We don't look for that, but I seriously doubt. But we don't look for that.
Okay. Well, very good. Christos, what's the best way for people to find out more from you? Where can they go?
Well, rppa.hu. I think they can find out everything from there. It's all in English.
Oh, okay, good. Excellent. Well, Christos, very good. Thank you for coming on the podcast. I appreciate it.
Welcome. Thank you very much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.